So we've got a nice table, types of microscope. In the spec it doesn't really say that you need to know much about the different types of microscope. However, there have been a couple of questions that sort of allude to parts of microscope. So here they are. Source of the image, so light. Light microscope uses light. So light rays, or photons of light. Light rays. Whereas these other two types use beams of electrons. So beams of electrons, and same thing here. Beam of electrons. How the focusing happens? Well, you've got lenses on the light microscope, a bit like your glasses. So glass lens. Whereas in these other two, they use electromagnets. You have to move because the electrons have a charge. If you use electromagnet, it kind of attract or repel a charge. So electromagnet. Magnification. So the ones that we've got here at college, the ones that you use go up to 400. Then we can put an extra lens in, an oil immersion lens that takes it to 1000. So 1000 times. Then it becomes very difficult to see things. We lose the resolution. And transmission electron microscope. For most images that you'll see, sort of times 500,000. You can get a bit more than that, but most images will be about that, up to that size. A bit less for scanning electron microscope. You tend to see things up to maybe 10 times less than that sort of size. Resolution. Low, or lower than the others. So we're looking at 200 nanometers. Remember, nanometers is 1,000th of a micrometer, which is 1,000th of a millimeter. So it's not bad, but for looking at really detailed stuff, you kind of lose the resolution. Whereas with our transmission electron microscope, then we can do 0.2 nanometers, so a lot better resolution, and slightly not quite as good for the scanning electron microscope, about 10 nanometers. Still pretty good though. Kind of live specimen we use? Yeah, you can put live things on your slide and under your microscope, see little hearts beating of water fleas and plants moving about and so on. So yes. But in the other two, the chemicals used and the slicing used, plating of specimens, it kills anything off. So no. Is it a section or external view? So you can do with light microscope both really. So you can cut sections and the light shines through, or you can. Small things, you can look at the external view by moving the focus. Transmission electron, that is definitely a section. They use an ultratome or a microtome to cut thin slices, like a bacon slice, but really small. So that is sections. And SEM, well, you can see the outsides of things, so it's the external view that you see because it plates the specimen. So external view. Cost. Well, you could buy. We let you use them, so they can't cost that much. So cost is low. Maybe you can get a decent microscope for, college microscope for £400, something like that. And the other two are very expensive. So. Cost is high. And depending on where you go, you might pay £100,000 for an electron microscope. So more than a Ferrari. Can the image be viewed directly? That's a bit of a strange question. I think that means, is it easy to look at it, so you can just stick it on and view it? So I think yes. And for these, no. There's a long process that you need to go through before you can see the specimen. And do we need to stain? No. But maybe. So if you're looking at a plant cell, you can see the cell, but you might stain it for, say, the chromosomes, or use some methylene blue or iodine stain, something like that. Whereas with the others, then you kind of need stains and coatings and things like that. I would say yes for the others. So I go yes, you need to stain that. Yes. Yes, or certainly coating of things, even if it's not like a traditional stains. And there's a Tyson microscope.